Our final speaker this morning is Heiner Wedemeyer, who's from Hannover, and he's going to speak to us on delta replication and putative drug targets.
Okay. Thank you for the introduction and also for involving me in this congress. This is the only talk on hepatitis delta, clearly the most severe form of viral hepatitis, and probably clinically the big remaining challenge. And therefore I briefly want to put some clinical information before addressing the primary goal of this talk, to discuss possibly novel drug targets. These are my disclosures, which also include companies being involved in the development of drugs against hepatitis delta. So you all know that delta uses HBs antigen as an envelope. It's really the smallest genome known to infect man. We have, importantly, no viral enzyme or no viral-induced enzymes, so there are no DAAs that can be developed targeting a specific HDV enzyme like proteases, polymerases. HDV uses host polymerases for replication, which I think is very important. The other point which needs to be highlighted in terms of drug development, that it has a very specific formation of HDV RNA, so it is highly paired. It has these rod-like structures, which makes it also maybe more challenging for simple strategies like, let's say, anti-microRNAs or antisense RNAs to be developed, because it's really this structure which is also difficult to be targeted. On the other hand, obviously, we have the components of the hepatitis B virion, and as Stefan nicely introduced to you, entry inhibition should obviously then also work against hepatitis delta, and I will come back to this in a minute. What also needs to be highlighted when we talk about hepatitis delta, that the delta enzymes and that the delta proteins, so the small and the large delta antigen, interferes with the HBV replication cycle. You have one virus infecting a cell, you have a second virus infecting a cell. The second virus depends on the first virus, so you need to maintain, delta needs to maintain some level of HBV replication, but not too strong to take up too much space. And therefore, there have been very nice studies clearly showing that, for example, the delta antigens reduce HBV replication, for example, by interfering with HBV enhancers, and this is a very well balanced, and this also explains why in delta infected patients HBV DNA levels are usually low. And I think this is important to keep in mind when you interfere with one compartment, that you then may have secondary effects also on the primary infection, which is delta, HBV. So delta is very diverse. We also have to keep in mind when we talk about new drug targets that we have 8 different delta genotypes. They are differentially distributed around the world. So you have genotype 3 in South America. We here in Central Europe and also in the Mediterranean areas see mainly genotype 1 infected patients. But we have, for example, in sub-Saharan Africa, genotypes 5 to 8, which is still an issue for diagnostics. So some assays miss some of the African genotypes, which has to be kept in mind. And in Asia, we have the genotype 2, which maybe is associated with a more benign clinical course. So this is also another issue we have to keep in mind. That delta is really more severe than HBV monoinfection is now very well established. This is the latest data published from the Greek cohort, a large number of patients showing that delta takes a more severe course. It is possible with clinical parameters to identify patients who have a more benign or more aggressive clinical course. There have been various studies. One was developed by a fellow in our department. She developed the so-called BIA score, where you can differentiate based on clinical parameters patients with 3 different clinical outcome scores. This course works in Hannover, but also with Maria Butti in Barcelona and also in Düsseldorf, and it's available on the website to identify patients who may need treatment more urgently versus patients where you may defer treatment for a a while. And there are other old factors which may be used in addition. For example, it's important to know that the majority of patients with hepatitis delta express anti-HDV IgM levels, and if these IgM antibodies are absent, then these patients have also more benign course. So you may go back to old diagnostic tools which still may have a clinical value right now in our practice. So what can we do right now if delta is common and if it's severe? Obviously, we have to be reminded that we have no direct viral targets addressing HDV RNA, and therefore we are left basically with interferon that has been used for 30 years in different studies, different readouts. Pegylated interferons have been explored. Together with geniodidine, we have been studying this in a cohort in Turkey, Greece, and in Germany. And the message is basically that interferon reduces HDV RNA suppression in about 1/4 of patients, and that in this respect, combination with adefovir was not superior in terms of HDV RNA suppression. However, it was superior in terms about HBs antigen suppression. This was the old trial with adefovir. The tenofovir combination trial has been presented last year at EASL. It was a 2-year treatment, so the aim was to extend treatment with interferon and to explore the combination with tenofovir. And here the message basically is that extending treatment does not significantly increase sustained response rates. So we are left with 1/4 to 1/3 of patients showing off-treatment HDV RNA responses. We also still observe, despite prolonging treatment, post-treatment relapses, which is an issue. And the interesting part here was that in contrast to combination with adefovir, we did not see a synergistic effect on HBs antigen reduction with tenofovir, which I think is still interesting. Why is it the case that with adefovir you observe this effect, but not with tenofovir? This needs to be studied. And the final point is obviously that in contrast to HCV, response to HDV with interferon treatment does not mean sustained virological response in terms of cure of the infection. So there can still be low level of viremia be present. You may have some latent reservoirs, which also has been shown in the post-liver transplant setting. And the point is you can observe late relapses in patients who have been HDV RNA negative 24 weeks after the end of therapy. So keep your patient on surveillance. The impact of these late relapses from the clinical perspective still need to be investigated in larger cohorts. So the current management then, to be summarized, is patients, I think, with a very mild course of disease can be identified. I showed you data on clinical parameters and anti-HDV IgM levels. Importantly, delta is also very well controlled by immune responses, and for example, monitoring NK cell responses can also identify patients responding to treatment, not responding to treatment. For time reasons, I cannot show you this data in more detail. At this stage, interferon remains the only effective treatment option. As I mentioned, however, long-term follow-up is required, and my recommendation would be to treat these BRB patients, so these intermediate disease course where you really may have a clinical benefit, while in those patients with a very mild course, you may wait for the better treatment options, which hopefully will evolve during the next years, and which I will show you in a minute. And finally, do not forget to treat hepatitis B. These patients are still infected with hepatitis B virus infection, and the important point is here to mention that some patients have HBV replication ongoing, even not that high as in delta negative patients, but reducing this very residual HBV viremia is believed to be associated with also more benign outcome. So, delta is bad, delta is not infrequent, and interferon does not really work. So what is on the horizon and what can we do? When you think about the delta life cycle, obviously we have HBV-mediated entry, as nicely shown by Stefan, and then we have different steps in the life cycle that potentially could be targeted in the future. Mercludex, Stefan showed you the data on the mono-infected patients. And in terms of co-infection, I only show you this one slide, this pilot trial, which again showed, like in the monoinfected patients, a beneficial effect in terms of ALT reduction. So this suggestion that hepatitis is mediated by immune attack of newly infected cells is also confirmed in the Delta study, which I think is striking, that you observe this in different cohorts. But also, and this is very interesting, that you have a reduction in HDV RNA levels. So in this pilot study, 6 out of 7 patients experienced this HDV RNA decline of more than 1 log at week 24 with Merclodex monotherapy. And if you combine this with pegylated interferon alpha, this was seen basically in all 7 patients in that trial. And I think we need to have more data, more trials are ongoing. It's really interesting that HBV entry inhibition may have an effect on hepatitis delta. Very interesting approach. What's about then targeting other steps in the HDV life cycle? And Amir nicely showed you the ideas of using gene therapies, and Stefan mentioned that it's now possible to perform high throughput screenings. I cannot show you yet any data. I expect many studies to be published in the next 12, 18 months for in vitro studies, screening for new drugs for Delta. All the tools that Stefan showed you apply also for hepatitis delta. We may see different molecules being identified in the life cycle, in replication, in translation, but yet there's nothing to be shown. But I think we might see something in the future, and Stefan may comment on this as a virologist during the discussion. What's about other steps in the life cycle? And prenylation has been investigated by Jeffrey Glenn for more than 20 years. He had a science paper published already in 1992. And what is prenylation? Prenylation is basically a lipid modification and post-translational steps of the HDV life cycle. And this blocking of this lipid modification leads in the end to interference with virion assembly, and it may also impact or block packing of viral particles. This is the idea which has been shown in vitro systems, and Jeffrey then published more than 10 years ago a paper in JCI in a mouse model also showing that if you block this step in this in vivo model, that indeed you can observe a viral decline. It then took more than 10 years to really perform clinical trials. And these first clinical trials have just been published. The proof of concept trial in Lancet Infectious Diseases went online, this paper, in July. This is a proof of concept study performed at the NIH in a small cohort of patients, so 2 groups of patients, and they received treatment initially only for 28 days. And you can see here the key figure of that paper. So it was a group that did not receive any treatment, and then there was one group using the drug lonafarnib, which is a prenylation inhibitor, at a dose of either 100 milligram or 200 milligram. And you can nicely see here that there is a dose-dependent decline, which started rather early during treatment. Already after 1 week, you can see this decline in HDV RNA. And what was also important, that there was a very nice correlation between lonafarnib concentrations observed in plasma versus viral declines during this first 28 days of therapy. So a very nice dose-dependent correlation of this drug, really suggesting a direct antiviral effect of this compound. And then there has been also some modeling of viral kinetics, which compared kinetics from interferon-treated patients versus patients receiving these lonafarnib drugs. What is interesting also, the half-life of HDV RNA, this was shown in a hepatology paper last year based on interferon-treated patients at the NIH, was at that time with interferon to be expected to be between 2 to 9 days, while with lonafarnib, the half-life was shorter in these patients. So there's also nice modeling now, which leads to better understanding of HDV life cycles, different steps important in this concept. So this was the idea. The problem was, obviously, at that stage, only 28 days of therapy, and the drug has then been used in Ankara with cyanididine, and he presented this data at EASL, where additional doses have been tested. So at the NIH, it was 100 milligram and 200 milligram, and here now cyanididine used also 300 milligram. And importantly, he also used combination with ritonavir, as the drug is metabolized via the cytochrome system 3A4. So boosting with ritonavir should be possible. And then finally, he also had 3 patients where they combined the drug with interferon, which also obviously is the next question to be addressed. You can summarize, this slide summarizes basically the antiviral efficacy at day 28 with all these different treatment arms of patients treated at the NIH in Ankara. It's small numbers, sometimes only 3 patients per dose group, but it's the very first proof of concept, getting a better idea for future studies which drugs to be used. You can see here that within 28 days, you observe about 2-log decline with a higher doses of lonafanib, and 100 milligram, which is the second bar from the right side, boosted with ritonavir, also observed this high response rates. So boosting with ritonavir seems to be interesting. How do the individual curves look like when you look this in these 3 patients, for example, treated with interferon? You can see here that there's a constant decline, and some patients received also in Ankara longer treatment than 4 weeks. Here you can see the 8 weeks data, which is important that you have a continuous decline after week 4 when you combine it here with interferon, which is interesting, and obviously we need now more data, longer treatment, and also better off-treatment response rates. There's always a but in life. This looks good. The but is the potential side effects. And lonafanib was associated with GI side effects. Many patients lost weight, even within 24 or with 28 days of treatment. This is certainly something which needs to be followed in future trials. What is the optimal dose for each patient? Will ritonavir reduce the side effects? This is ongoing work, and more patients are currently studied in various small trials, and we have to simply wait whether the side effects will be dose limiting. The last very brief data step I want to show you is another idea, interfering again with the HBV life cycle and maybe blocking subviral particle formation. And it has been suggested that nucleic acid polymers would not only work in hepatitis delta, but also in HBV monoinfection, but also in hepatitis delta. And this trial has been presented at EASL, patients treated in Moldavia. You can see here that the REP 2139 drug was given IV for 15 weeks in hepatitis delta patients. You can see here a dramatic decline in HBs antigen using this approach within starting basically after week 2 or 3, and then you see this decline with 2 different groups of patients having more rapid decline versus a slower decline in HBs antigen. And very importantly, this was almost paralleled by a decline also in HDV RNA. And if you would then plot S antigen decline versus HDV RNA decline, you can see here these 2 groups of S antigen declines versus also 2 groups in HDV RNA decline. Something needs to be followed in terms of toxicity of this approach. ALT increases were not reported, etc. I think very interesting data, but we need to be cautious and have to wait for more data of this approach. So in summary, I think at this stage, PEG interferon remains the only treatment option for delta. Both entry inhibition, prenylation inhibition, and maybe block of particle formation may represent future treatment options, and novel strategies to achieve HBs antigen clearance, as Amy nicely showed to you. I think they also need all to be explored in hepatitis delta. If you get rid of S antigen, I don't need a specific treatment maybe for hepatitis delta. And if you want to be involved in delta, I invite you to join our efforts of the hepatitis delta network, and more information is found on that website. Thank you very much.